Hello, this is Dr. Michelle Plowman, Associate Professor and Canada Research Chair in Neuroplasticity, Rehabilitation, and Brain Recovery. Welcome to the Recovery and Performance Laboratory, located at Memorial University in St. John's, Newfoundland and Labrador, Canada. Our laboratory is uniquely situated within the province's tertiary rehabilitation center, the Miller Center, so we're easily accessible to patients and clinicians. We focus on creating and testing innovative treatments that promote neuroplasticity and restore lost functions after injury to the brain, specifically in multiple sclerosis and stroke. Our laboratory is equipped with state-of-the-art technology that can probe how the brain controls movement. For example, here Lindsay is demonstrating the use of transcranial magnetic stimulation, or TMS. TMS uses a magnetic pulse delivered to the surface of the brain to measure the speed and quality of brain circuits. We are now using TMS to map the connections from the brain to the hand. People with MS are concerned about problems with hand dexterity, which interfere with tasks such as typing, cooking, and dressing. So we use TMS to try and figure out what circuits to target when we create new therapies for hand impairment. Bruna is a master's student who's investigating a new way of measuring mental fatigue. People with MS tell us that their fatigue limits their ability to work and maintain their personal obligations. Funded by the Canada Foundation for Innovation, the Functional Near-Infrared Spectroscopy System, or FNIRS, measures the amount of oxygen use in the brain. We think that fatigue could be related to inefficient use of brain resources, which we can measure using FNIRS. Our future research will disentangle and then create new treatments for mental fatigue. Michael is a master's student in the lab. He uses a sophisticated robotic unit called KinArm to measure the recovery of the arm and hand after brain injury. The robotic actuators provide a realistic virtual reality environment in which the person interacts. We can measure with amazing precision sensory and motor integrity of the arms and the hands. This unit will be able to detect improvements with new therapies that we will develop aimed at improving and restoring arm and hand function. Because of our unique focus on neuroplasticity and recovery, we are contacted by people with MS and stroke from all over the world. In collaboration with Drs. Mark Stefanelli, Fraser Clift, and Craig Moore, I have spearheaded a unique and potentially impactful project that maps the cognitive, physical, emotional, and immunological health of people with MS for 10 years. This project, called the HIT-MS Project, involves annual visits to our laboratory of about 200 people with MS. In time, we'll be able to understand the factors that protect against progression of symptoms in MS. We've been funded by the MS Society of Canada to conduct a trial of our Cool Room bodyweight supported treadmill method to restore walking in MS. In this trial, we compared two versions of the therapy to determine which one more effectively stimulates neuroplasticity and improves walking. We use a special instrumented pressure-sensitive walkway made by Protokinetics. When the patient walks across this walkway, the load cells on the mat are activated, so we can detect subtle changes in walking patterns in response to treatment. This technology detects changes that are happening, not seen with the naked eye. Research such as ours requires funding. We compete for grants from agencies like the MS Society of Canada in order to support our projects and to provide study fellowships for master's and doctoral students. Investing in these students is an investment in the next generation of researchers and research-minded clinicians. Thank you for visiting us in our lab. Consider supporting research like ours that helps repair the brain and improve quality of life.